Okay, so I would also like to start by thanking Hugues for inviting me today, and I should also make a declaration maybe or disclosure right away, which is I'm not a DNA damage person. I worked mainly all my life with mitotic cells, with events like this, is better. With events taking place in the cytoplasm. So we're just new to this field, and so I'm very grateful to be present today so that I can get your feedback, and I have learned already quite a lot. So as you probably all know, most cells in our body are diploid, meaning that they contain 2 copies of all chromosomes. However, there are exceptions to this. If they contain 3 copies, they will be triploid. In 4 copies, like in this example over here, they will be called tetraploid. And this is really an interesting concept, because as whole genome duplications and polyploidy, when the whole genome is present in more than 2 copies, have actually been used throughout evolution to generate a few organisms like fish and certain frogs, but also by man, so that tomatoes get bigger or strawberries, and even oysters that you can eat them from the French coast, Atlantic coast in the summer. They will most likely be triploid. So in our bodies and in the bodies of many animal organisms that we use, on top of having diploid cells, we also have polyploid cells, and I like to call your attention, for example, for megakaryocytes, who will be increasing in size and in ploidy so that they can produce a lot of RNAs and proteins that will then be used in the formation of platelets. But there are many other examples, such as keratinocytes or cardiomyocytes. In these examples over here, so the so-called programmed polyploidy, it's always advantageous to actually gain these functions through differentiation, so that these cells can now increase metabolic capacity or have a barrier function. But with whole genome duplications, we realized that with the deep genome sequencing, we realized that whole genome duplications are actually present in a variety of human tumors and associated and correlated with high levels of genetic instability. In a key experiment in the Pellman lab, published 20 years ago, it has been shown that cytokinesis failure, so now a cell will be formed with 2 nuclei and so double the amount of DNA, in a p53 null background, this will actually be sufficient to generate tumors. So the question we started to be interested a few years ago is to understand how non-programmed whole genome duplication can actually lead to genetic instability and maybe be causative of tumor development or progression. And so we fall into this completely by chance when a student in the lab was working with neurostem cells from the fly brain. They are called neuroblasts, and she realized that cytokinesis failure, which normally should lead to a p53-mediated arrest, in this model system was just not causing any cell cycle arrest, and cells will just continue to proliferate and repeat cytokinesis failure. And she also found out that this was actually already known, as we were discussing over lunch, that sometimes people have already found out quite interesting and important things. And so you can see that instead of being diploid and small like they normally are in control wild-type brains, they become these huge monsters that still express a neurostem cell marker and just keep proliferating, completely invading the diploid counterpart in the brain tissue. So Madalena Nano, the PhD student I was just mentioning, study what happened to these cells as they fail cytokinesis many times, and she found this very strange phenotype where, by looking at gamma H2AV, the homologue of gamma H2AX, which labels double-strand breaks, in mitotic cells, there were regions where we could see high levels of gamma H2AX or V, but it was not distributed in the entire nucleus. She started filming these cells. So this is a very large neuroblast that has failed cytokinesis many, many times. And when the movie will start, if we look at the localization of RPA, which labels single-strand DNA, we will see that a few nuclei will actually very rapidly and transiently accumulate foci of RPA that then will disappear. So if I show you the same movie now, looking at also histone RFP, you can see that the nuclei that accumulate this green signal, the RPA signal, are actually delayed in mitosis and not condensing their chromosome as the other remaining nuclei. And so here are the stills so that it may be easier to see. Now, this reminds us, here is just the controls showing that if we laser ablate diploid neuroblasts, we'll see RPA actually accumulating, and we're sure it's nothing to do with our filming conditions because we can film these cells for many diploid cells for many consecutive mitoses, and they never accumulate RPA foci. Now, we did the same experiment in human cells. These are ovarian cancer cells where we induce cytokinesis failure. In this case, they have three nuclei, and these two are ready to go into mitosis, not this one. And as these two go into mitosis, this one starts fragmenting and forming this kind of very small DNA pieces, and then the cell divides, and these remnants, kind of micronuclei behaving, will stay in the cytoplasm. Now, this, of course, reminded us of experiments done 50 years ago where fusion of HeLa cells that will be in S or in M phase will actually impose DNA breakage, what was called at that time by Rao and Johnston, DNA pulverization very rapidly. So we wonder if the problem was related with cell cycle asynchrony. Maybe these nuclei are really not progressing in the cell cycle in the same way. And this is pictures. These are pictures of control planes diploid. You can see each nucleus has a different color. There's a lot of green because there are neurons at this stage of development already differentiated. But you can see that in the polyploid brains, then we have these very large shells full of different nuclei, and some of these cells really show a high asynchronous nuclei population. So we try to synchronize these very large shells by treating them with a CDK1 inhibitor and then release, and indeed we can actually see them going into mitosis with less DNA damage when compared to controls. So we learned that if a cell is not meant to be multinucleated, and we have examples of multinucleated cells in our body, like hepatocytes or muscle cells, but in this case we know that these cells should not be multinucleated. One of the problems is that it will show DNA damage at mitotic entry, which will probably have drastic consequences in terms of genome integrity, and that multinucleation can lead to cell cycle asynchrony, which will generate mitotic damage, DNA damage at mitotic entry. Now, while Madalena was studying the behavior of these chromosomes during mitosis, Alex Kupil, another PhD student in the lab, also joined this project, and they realized that in sharp contrast to diploid neuroblasts who divide in an asymmetric manner, generating a neurostem cell and a differentiating progenitor, and they always divide in a bipolar manner. When we characterize polyploid Drosophila neurostem cell mitosis, we could see that the extra centrosomes and chromosomes seem to not be coordinated, so that there is an initial clustering of the centrosomes, but then the spindle fails to basically elongate and scale, and it will be very chunky and tubby and divide in a multipolar manner always. Now, we are also interested in the lab in centrosome amplification, so the phenomenon by which more than 2 centrosomes are present in a cell. And we and many others have shown that cancer cells actually have very robust mechanisms to make these extra centrosomes to cluster and form bipolar spindles, so to minimize aneuploid cell generation, at least by the progeny of generating 3 cells. But now, is the movie going now? If we induce cytokinesis failure, if we induce cytokinesis failure in these cells, we can see, the movie is not playing now. Thank you. We can see that now we see exactly the same as in Drosophila. The cells will divide in a large majority in a multipolar manner. So with this, we learn that mitosis, when you have extra chromosomes and you're not meant to have so many chromosomes, it will be problematic. And so we wanted to go one step before and see how would be, what will be, how the first interface of a cell that it's not supposed to have double the genome will occur. And so this was part of the work of Simon Jambol, a postdoc in the lab, and he decided to answer to this question using human cells in culture, and in particular RP1 cells that are diploid or almost diploid, genetically stable and untransformed. And Simon generated several ways of inducing either cytokinesis failure or mitotic slippage through endomitosis or endoreplication. And so the differences in this case is that through endoreplication, there will be an alternate S and G phase, generating double the DNA content, but without going through mitosis. While during mitotic slippage, these cells will enter mitosis, will even condense chromosomes, but then will exit mitosis without anaphase, so they only have 1 nucleus. And in cytokinesis failure, they will generate 2 nuclei, as you can see over here. And so throughout my talk, I will show this kind of graphs where we always have a control. For example, if we induce cytokinesis failure with an AID system that targets MKLP1, there will be in the same well diploid and tetraploid because our treatments are very short, but then there will also be another control where there is no auxin. And so we can identify by counting the number of centrosomes and the nuclear area, tetraploid cells from diploid ones. And independently of the way we were generating tetraploid cells, we could see an increase in gamma H2AX within the first interface. Now, of course, we wanted to understand when this was happening, and so using the Fuji system, we can follow precisely at which stage of the cell cycle these cells are while measuring DNA damage levels. And we can see that as they are born as tetraploid cells, there's very few DNA damage levels in the first G1, but as they progress to S phase, this actually increases. And so if it's generated during S phase, is it generated through DNA replication? As we heard this morning that it's very common the case. And indeed, it seems to be the case because if we use very, very low doses of afidicolin so that cells are in S phase with a biochemical environment of S phase, but forks are progressing very slowly, we decrease the levels of gamma H2AX. There was still this population of cells that was actually perturbing me, and also you can see them here on the graph. And so Simon repeated these experiments by also revealing the levels of EDU, and indeed there is a high correlation, as you can see over here, with the levels of gamma H2AX and EDU. Now, since there were these defects in S phase, we wanted to understand how this will lead to genetic instability, in which way this will lead to genetic instability. And so we characterized the level of genetic instability in G1. These are single cell DNA sequence. Each line is a cell, and here you have the chromosomes, and you can see that, as expected, karyotype of 4N. And if we now look at cells that have been through S phase, are in G2 just before going to mitosis, most of them are green. Again, karyotype expected, 8N, but you can see there are large regions that are under or over duplicated, really showing us that just one S phase was sufficient to generate these errors in DNA replication. Now, at this time, we start looking at DNA replication by looking at pCNA behavior, and we realize that there were actually that pCNA was actually behaving very differently between diploid and tetraploid cells. I hope you can see the salt and pepper behavior. Maybe it's easier like this. So while in diploid cells, very rapidly pCNA reaches a plateau, it doesn't stay for long in this plateau, and then the number of foci decreases also very rapidly. This is not at all the case in tetraploid cells, and if we count the number of foci in a diploid cell that is undergoing DNA replication and in a tetraploid cell, you can see that it doesn't go to double the amount, which is exactly what we were expecting. So we did mass measures, I'm not showing here, and we had this surprising result that indeed the mass accumulation between diploid and tetraploid cells was not double as expected. And so we start looking at DNA replication components. And so what we did is, from the same culture, we FACS sort diploid and tetraploid cells, and then do protein extracts by lab and migrate the same number of cells on protein gels and perform Western blots, either total protein extracts or nuclear extracts. I will show you in a minute. And you can see that if we normalize these DNA replication proteins, or E2F1, which is a transcription factor that it's involved in transcribing several cell cycle regulators, when we normalize by H2B signal, which is an histone marker, you can see there's not a true scaling as expected. And this is also true from nuclear extracts. So we wanted to compensate for this lack of scaling, and so we overexpress E2F. And indeed, now we can see a partial rescue of DNA replication factors. We also look at cell cycle because we were wondering if at G1S transition there's not enough mass. Maybe a possibility was that G1 is not taking long enough to be able to accumulate this mass. And this isn't really the case because, again, even if statistically significant, you can see that G1 it's not very different between diploid and tetraploids. So using a way of inducing a G1 delay with palbociclib, we could actually rescue the level of DNA replication factors in E2F1, which lead to a different behavior in terms of DNA replication and DNA damage. So this shows us that newly born tetraploid cells really cannot count their number of chromosomes. This leads to a lack of scaling up in terms of protein content and cell cycle timing and DNA replication defects, which will then maybe have a condition in the way these cells will actually proliferate. Now, we wanted to understand if there are factors that one could identify that become essential for polyploid cell proliferation. And Manon Budzik, a PhD student in the lab, decided to do a screen for factors involving DNA proliferation. And so she screened using the FLY system again, because it's quite cheap, where we can have these polyploid neuroblasts that are very large, where their cell area correlate quite well with nuclear area, and do an RNAi screen to identify factors that produce now polyploid cells that can no longer proliferate and so should be much smaller. And this is what she obtained, a few of those, like the ones in green, that become smaller. Some of these nucleases, like I will be talking about Gen over here, but others such as FEN1, which is from the same family, but also quite surprising others where actually cells became bigger. So Manon decided to focus on Gen 1 because Gen 1, or Gen in flies, but Gen 1 in human cells, it's kind of this strange nuclease that it's localized in the cytoplasm. It's mainly been studied by the lab of Steve West at the Curie Institute, and he has shown that Gen 1 is actually maintained in the cytoplasm through a retention sequence. Now, we know that Gen 1 is involved in the resolution of homologous recombination products, and it's also, you can see, highly associated with junctional DNA, as you can see over here. So homologous recombination will be using, as you probably all know, RAD51, and these RAD51 filaments, and this will be generating holiday junctions that have to be resolved, and they can either use a BTR pathway, mainly using the BLM helicase, or a mix of a scaffold with other subunits on nucleases, SLX4, SLX1, and finally Gen 1. And basically, when you look at the number of papers on Gen 1 and on the other ones, it's really not comparable. There's really very few studies on Gen 1 because it's always this kind of strange nuclease that only comes at the very end, and it's really not necessary even in mouse mutant models. So it's kind of, there's always this idea that it's not really an important nuclease. So we decided to compare the depletion of Gen 1 with the depletion of the other nucleases and with RAD51, again, still in the same system. And we were very surprised to see that while RAD51 gives these defects in proliferation as expected, because without homologous recombination, these high levels of DNA damage will probably be not compatible with proliferation, none of the other proteins, BLM or MAS81 or SLX1, were actually involved in DNA in proliferation of polyploid cells. And Gen 1 here, once more, we could see exactly the same behavior. Now, while we were starting to be interested in Gen 1, Steve West also published this paper showing that Gen 1 also promotes common fragile site expression. In experiments done in HEX cells, it shows that in cells treated with very low doses of afidicolin, actually cystochromatid gaps that are typical of control cells, let's say, will be reduced when Gen 1 is knocked out, or MAS81. And if you do now both, if you remove both, this will not increase, suggesting that they are working exactly in the same pathway. And here you can see an increase in the number of bridges, DNA bridges, which is compatible with the lack of fragile site resolution. So still in flies, we realized that Gen 1 in flies works exactly as in human cells, but we can also see it associated with the nucleus, actually, even before nuclear envelope breakdown. This is a movie showing exactly that, where there is already fluorescence inside the nucleus before nuclear envelope breakdown. As you can see over here. And to our surprise, if you look at the Gen mutant, it's viable fertile with a few mitotic errors, but nothing really major. And if we overexpress Gen, it's also viable, although cells spend more time in mitosis, just like in the mutant, and there are a few chromosome defects, chromosome behavior defects during mitosis, but not a huge increase in DNA damage in the diploid condition, so in the mutant or overexpression. Now, in contrast to this, in the polyploid background, these cells stop proliferating. This is confirming the RNAi screen as you can see over here, while the overexpression is making these cells much bigger. So from a genetic point of view, this is quite interesting because it's showing that the depletion of gen abrogates polyploid cell proliferation, while the overexpression is basically promoting. So we learned that gen and RAD51 are absolutely required for non-programmed polyploid cell proliferation, that gen depletion impairs the proliferation of polyploid cells, and that gen overexpression favors polyploid cell proliferation, but we do not know from the fly work if it's HR dependent or fragile site resolution, or can even be both. So what happens in human cells? So again, we look at the levels of gen. Remember about the lack of scaling up. Actually, we were surprised to see that in RP1 cells, in newly born tetraploid, gen 1 is actually almost scaling up, as you can see over here, when normalized by H2B levels. And then we spend a lot of time generating gene knockouts in RP1 cells and also in cancer cells in HET116, which are P53 competent, but still cancer cells with quite stable genome content. And we have indeed a few cell lines that either by knocking exon 2 or 3 show very low levels of gen 1, as you can see over here. So what we do is that we induce tetraploidy in these cells, and then we release them, and we characterize the first cell cycle after a whole genome duplication. And we can see that there is a very slight increase in the number of gamma H2AX foci after gen 1 knockout. Not much differences in terms of RAD51, which I guess was expected. Gen 1 is downstream. If we compare tetraploid cells with gen and tetraploid cells without gen, but a substantial increase in FuncD2, which goes along with the lack of fragile site resolution. Now, in addition to this, we can see defects in mitosis. Actually, there are even more multipolar mitosis when gen 1 is knocked down. Here it's a mistake, it's not SH, it's the knockout. And also in the increase of ultrafine bridges and pitch positive bridges. So we can also see that gen 1 loss results in increased gamma H2AX levels in mitosis, which was kind of surprising, but maybe goes with really the fact that these cells are entering mitosis and either they still have damage or they are generating more DNA damage. And what will be the subsequent G1 will have an increase in 53 BP1 levels. We then wanted to understand if we could we wanted to know why these cells were no longer proliferating in the fly, if we could actually see the same in human cells. And so we turn again to the Fuji system, and while, again, this is all the knockout cell, we also have the SH, but I don't know what happened here. We can see that indeed from without gen, diploid cells go through the cell cycle. With gen, tetraploid cells also go through the cell cycle as expected, but without gen, we'll start seeing something that was completely unexpected, which is they will spend more time in S phase, and then they will not enter mitosis, but they will actually go directly to G1. This is called mitotic bypass. It's basically described in cells that have high levels of DNA damage or even high levels of cyclin E. And so these are the profiles of the cell cycle. You can see that there's an alternate cell cycle between SG2, M, and G1 in diploid, and also in Gen 1 diploid. So somehow there are cells that spend a lot of time in G1, as you can see over here already, in diploidy, but really not as much as we can see in tetraploid cells, where basically cells are really losing proliferative capacity. Now, we turn to the HET116 to actually see if we were seeing the same, But we realized that even if there is more gamma H2AX foci, the levels of RAD51 in this case were also increased, but not FUNC-D2. So it's really quite different from what we were seeing in RP1 cells, but also the levels of 53 BP1 in the subsequent G1 phase after the first mitosis. And interesting enough, we do not see that Gen1 loss can actually induce the same level of proliferation arrest in these cells. Now, in both cases, P53 is present, and if we remove P53 from RP1 cells, they will keep going. So a prediction of our findings is that a tumor that has tetraploid cells will be relying on Gen1 to continue to proliferate. And so we team up with Isidro Cortes at the UBI near Cambridge, and he analyzed a series of tumors where we have the annotation for the levels of whole genome duplication. And indeed, we can see that several tumors actually show an upregulation of Gen1 when they are whole genome duplicated. So we're conceiving this way of looking at tetraploidy where a few proteins that are actually apparently dispensable in diploid conditions show replicative stress, and this makes them maybe more vulnerable to deal with genetic instability. So let's say in a tetraploid cell where gen is present, high levels of DNA damage will lead to genetic instability. This may or not lead to a winner karyotype that will then carry on. But if there is too much DNA damage because Gen1 is not present, these cells will lose proliferative potential, and then this will lead to, due to higher levels of fragile site and defects in HR resolution. And so we also think that it's interesting to look at gen because apparently it may facilitate cancer cell proliferation through upregulation of its levels. So this is it. We're still trying to understand more about Gen1, but I would just like to again mention the name of the people that did the work. Most of it was done by Manon Boudzik, with also some help by Anthony Simon, who is our lab manager and who generated the CRISPR knockout cell lines. And I should mention also Steve West, because we try to generate antibodies and we, I don't know, spend a fortune in buying antibodies and generating our own, and they never worked. And Steve has been very generous in giving us a lot of Gen1 antibodies. And with this, I thank you for your attention.